So thank you very much for that kind introduction. I'm going to tell you about some work we've been doing on the intracellular transport, and Jacques Pro gave a very nice introduction last night. Let me just briefly review it. Of course, you've got over 200 different kinds of cells in your body: blood cells, muscle cells, nerve cells. And cells are not just bags of protoplasm, but highly ordered. And in many ways, I like to think of a cell as a city. It has all the infrastructure that you find in a city. There are proteins that are workers. Mitochondria are the refineries or power plant. The transportation system consists of actin filaments and microtubules. The trucks are kinesin, dynein, and myosin. Motor proteins, ribosomes are the factories, the genome, the library. Recycling centers, the lysosome. Police are the chaperones or ubiquitinating complexes. The post office is the Golgi, where things are packaged, labeled, and shipped. And communications occur by chemical signaling networks. So I'm going to focus on this transportation system, roads and trucks. And inside a cell, small molecules like ATP can move by diffusion, but larger things move by active transport of shipping cargoes. And it works a lot like the container shipping industry. So here is a artist's rendition with the microtubules coming away from the nucleus as these yellow fibers and the cargoes are blue. And the microtubules are like the interstate highway system. The minus end is anchored near the nucleus, and the plus end is out near the periphery. And motor proteins haul cargoes back and forth along these filaments. So these motor proteins that move the cargoes along the filaments, here's a rendition of, say, a bead or a vesicle with many motors on it, and just a few are actually doing the active transport. Kinesins would move toward the plus end outwards, and dyneins would move on microtubules inwards towards the nucleus. So the roads are the actin filament and the microtubules. The actin filament has a 74 nanometer repeat distance that is matched by the step size of motor proteins. The microtubules consist of 13 protofilaments arranged in a cylinder with an 8 nanometer repeat distance that's matched again by the step size. And so, as I said, the microtubules, which would be the white lines, act like the interstate highway system. The red lines would be like the actin, the local surface streets. And so the motor proteins walk along the actin filaments just like you do. They put one foot in front of the other. And this is myosin 5 walking along actin, and its distance between these feet or heads is 37 nanometers, matching the repeat distance of the filament. And so that's been as measured by Paul Sullivan's group. These are the three motor proteins. Kinesin walks along microtubules for the plus end, 8 nanometer step size. I showed you myosin in the last slide. And then dynein is a little more complicated. It walks along microtubules towards the nucleus. Steve Gross's group has recently found that it actually has a gear shift for transmission, so it can take 8, 16, 24, 32 nanometer steps, depending on the load and the ATP concentration. So these transportation systems are very important in the health of your cells and nerves, and it breaks down in neurodegenerative diseases, such as, and so you see a breakdown of this transportation system in things like Alzheimer's, Parkinson's, Huntington's disease. And we don't know if it's the cause or effect, but we know it's there. So here is a movie of herpes virus moving along microtubules inside the axon, which is a long filament of your nerve fiber, and it's moving along microtubules. You see some going backwards and some going forwards. And viruses often invade and co-opt the transportation system inside a cell. Okay, so the big question in this field is, how does a cell regulate the transport of cargoes? I'm not going to answer that big a question, but I'll show you a little bit of one thing we've done, which illustrates some of the ways to think about this. In particular, experimental colleagues of mine took, you take a frog or a fish and it sees a predator and it might get eaten, so it gets scared, so it changes color. How does it change color? Changes color by having pigment granules, say, gathered at the center of a specialized skin cell, pigment cell, and then the transportation system spreads these pigment granules over the surface of the cell. And that spreading out we call dispersion, and then when it wants to change back to the old color, it pulls those granules back in toward the center, and we call that aggregation. So here's a movie of granules dispersing outward from the nucleus. Each granule is about half a micron in diameter, and the cell itself is about 70 microns in diameter. So they've been given a chemical signal, and now they will disperse outwards.
How big is a granule?
Half a micron in diameter.
How long did that take?
This is sped up several times, but usually it takes about 200, 300 seconds, so several minutes for something like that to happen. This is aggregation where the granules have been spread out.
What would be diffusion time for the same?
Huh?
What would be the time for diffusing to the same distance?
It's complicated. I can't figure that out in my head because it depends on the size of the particle diffusing, and there are all the actins getting in the way and things like that. But it would be longer because it's only 40 nanometers. Okay, so that's aggregation where the granules go back in. And so what we did was these colleagues of mine took these pigment cells. They got rid of the microtubules by chemical treatment, leaving behind only the surface streaks, the actin filament. They took movies and tracked the pigment granules, the melanosomes, and they recorded the displacement versus time. So displacement is as the crow flies. And they plot the mean squared displacement, R squared average, versus time. And this is what they get. This is the displacement squared versus time. This curve here is dispersion. The lower curve is aggregation. The fact that you see a straight line means that a random walk or diffusion is taking place, just going on this random actin network. And the question is, why is this curve above that curve? Why after 50 seconds do you go further during dispersion when you're spreading out than during aggregation when you come in? You might think that the answer, maybe they're walking faster during dispersion. The answer is no, they walk at the same velocity, so that's not the answer. The answer turns out to be that during dispersion, the cargoes go straight to the end of the filament. They don't turn at any intersection, and therefore they go further, and this is a good way to spread out the pigment granules. On the other hand, during aggregation, there's a 50-50 chance of turning at one intersection or another, and if you have a 50-50 chance of turning at an intersection of two roads, you won't go as far, and this may be a good way to do a local search to find the nearest microtubule to go back to the nucleus. So let me just sketch how we came up with that conclusion. This is what we've been able to show theoretically. One thing we did was we took an equation that interpolates between short-time straight-line motion and long-time diffusive motion, and we fit the data to that. The other thing we did was Monte Carlo computer simulations of cargoes walking along filaments. So as I said, the first thing we did was we took an equation that interpolates between short-time ballistic motion and long-time diffusive motion, and that equation turns out to be the solution to the Langevin equation. And from fitting the data, we can get the mean free path, which is the distance between turns. So here's the solution to the Langevin equation. Mean square displacement is linear in time at long times, and it has two fitting parameters, the diffusion constant d and the time tau between turns. And from those two fitting parameters, you can get the mean free path. Take this product and the square root. So this red line here is against mean square displacement versus time. The red dashed line is the Langevin fit, and from that fit we get two mean free paths, 539 for dispersion, 237 for aggregation. And we then have, this is the electron micrographs of the actin filaments artificially colored in yellow, and the lengths I showed you are consistent with having the dispersion about half the length of the microtube of an actin filament, so it gets on average in the middle, goes to the end. The other mean free path is consistent. It's 1.5 times the distance between intersections, and it's consistent with a 50-50 chance of switching. So these look denser during aggregation than during dispersion. We don't have enough to quantify that, but we can sort of get a measure of that density by plunking down little circles whose diameter is the diameter of a cargo, and then counting the number of yellow filaments going through the circle, and we call that the touching number, and it's about 8 for aggregation and 4 for dispersion. And then we use that in Monte Carlo simulations that we do. These gray lines are the actin filaments, the colored lines are the trajectories. And we vary the filament length, the density, and the switching probability. And here is the result of the simulation, the mean free path versus touching number. And for the numbers I gave you, the touching number of 8, the mean free path on the y-axis of 237 is this gray. It intersects at the purple line, which is a 50% chance of switching. Similarly for dispersion, 4 is a touching number, 539 dark line mean free path, and they intersect at the red or green, and that's a 0 to 6% chance of switching. So basically, it goes 0% chance of switching goes straight through the intersections for dispersion, 50-50 chance of switching during aggregation. And to summarize, then, we see active transport of cargoes along filaments, and the cell can regulate the switching probability at intersections. So currently what we're working on is how does the switching occur? How does the system change the switching probability? We had a hypothesis that maybe there was a tug of war, some motors attached to one filament at an intersection, other motors on a cargo attached to the other one, and they kind of duke it out. That doesn't look like it's going to work. It doesn't give you enough of an effect. And what we think is going on is that there are regulatory proteins attached to the filaments and also activating or deactivating motors that can regulate the switching probability. So here are my collaborators. Joe Snyder did the work, and Steve Gross is the biologist who told us about this problem, and these are the experimentalists who did the work. Thank you very much.
Questions? Thank you.
Yeah, maybe I missed something. But your model is ballistic for time shorter than the times between turns, right?
Right, because it's walking on the filament.
diffusive the other times. But on the way out, it's also always when they go straight, they are always diffusive. You said.
No, that when they're going on the filaments, they're just ballistic.
When they go out on the filaments, they're ballistic?
Well, we got rid of microtubules, so they're just kind of randomly going on this random network. They're just going straight up line.
I thought you said that both way out and way in are diffusive, according to the...
They're diffusive because, long times, because they switch from one filament to another. They go straight along the filament, fall off the end of one, or whatever, go to another filament, go along that.
I thought the difference between going out and coming in was that in one case they go straight, and when they come back they switch between filaments.
No.
Okay, maybe I understand.
They go on the filaments, and long times they're diffusive in both cases because they're switching active filaments.
So they're switching both when they go out and the way.
Yeah, but in one case it goes all the way. You're right. Dispersion goes all the way to the end of the filament, falls off the end, goes to a new filament, goes all the way to the end of that one, falls off. So in that way it does go straight.